And I will present you the second speaker of this session, Dana Rubi Levy. Dana did her PhD in the Department of Neurobiology at the Weizmann Institute of Science, where she studied the neural correlates of social behavior and autism. Currently, she's a postdoc at PopData Lab at Harvard Medical School, where she studies sex-specific and inter-individual behavioral variations, as well as the evolution of behavioral patterns across life in mice. In today's talk, she will present her research on how individual identity and not estrous state diversifies the mouse spontaneous behavior. Let's please give a warm welcome to Dana.
Hi, everyone. I'm going to try to use this. Thank you for having me here today in this important meeting. Thank you, Danielle, for a very kind introduction. So today, sorry, I changed the title of the talk a little bit. I want to talk to you about biological sex, individuality, and the importance of behavior variation. So behavior variation, which is my field of study, seems like this very broad scientific term, but we actually all encounter it in our everyday lives. So here is an example from my everyday life. This is an actual image of my dining table after my kids have done eating. So I have two kids, a boy and a girl, a male and a female. This is their biological sex as assigned at birth. My daughter is a little bit older than my son, so they're of different ages. And ever since I can remember, my daughter has been the messiest kid you will ever meet. Well, my son is very tidy and clean. And so all of these are sources that can affect the variability in the eating behavior that you see on my dining table basically every day. And there are, of course, many factors that can affect behavior variability. And as scientists, in order to make sense of our data, to interpret our findings, we many times try to decrease variability in our data. And there are many ways in which we do it. We, for example, average over individuals. And we only look at adult subject, or we choose one biological sex to work on. And when we do that, we create biases. And actually, one of the most prominent biases in research is the sex bias, because for decades, males have been the default sex to work on in basic research and in preclinical studies. And females were very, very much underrepresented. So, and this is true across all biomedical fields, but I'm a neuroscientist, so I'm going to show neuroscience as an example. Neuroscience is actually one of the worst. So in 2009, about 15 years ago, a landmark review study was published. They reviewed hundreds of neuroscience papers and found that almost 50% of these animal studies, they only studied males. So they only used male subjects. And as I said, this is true across all biomedical fields. And the bias was so bad that a few years ago, the NIH, which is the main sort of funding institute in the US, decided to mandate the use of both sexes as a condition to get funding. And we do see this beginning of a positive trend of being more inclusive in studies, but the bias is still very much there. So this is a review from 2020, again, neuroscience, animal studies, showing that still 30% of the papers are only working on males, are only studying males. And this such strong sex bias naturally translates into a bias in the diagnosis and in the treatment of women. So again, just a few examples. In the UK, women are 50% more likely to be misdiagnosed during heart attack. In the US, 33% more likely to be misdiagnosed during strokes. Drugs do not work as well on women, so they suffer more from chronic pain, but morphines are not as effective. And overall, women suffer more from adverse effects to drugs. And so the question is why? Why would scientists choose to only work on males, or mostly work on males? And the reason lies in behavior variability and in the female reproductive cycle. So human females, we have a reproductive cycle of 28 to 32 days. Female mice have the estrous cycle, so that's the reproductive cycle. It takes about 4 to 5 days. It consists out of 4 different stages. You don't have to remember those. I'll refer to them during the presentation as their first sort of letter, and I'll use different colors. You really don't have to remember it. But there are 4 stages, each one lasting a day, and they're accompanied by fluctuation in hormones. So basically for a female mouse, each day, the hormone levels are different. And this is what led to the assumption that female data is much more variable than male data. It's noisy. It's uninterpretable. We cannot work on females. We won't be able to figure out the data. And this is one of the main sort of ideas or assumptions that facilitated this sex bias. And it is true that the estrous cycle and hormones affect many systems in the body. They even affect behavior. They affect sexual behavior and social behavior, reproductive behavior. They're supposed to, that's biology. But does the estrous cycle really affect all other behaviors that we as neuroscientists study? And even if they do they really diversify female behavior more than male behavior? Do they make female behavior more variable? And so even though there's a lot of literature on this, there isn't really a definitive answer. And so our study decided to target directly these two questions by using a very straightforward approach. So we studied the behavior of 16 females over 15 consecutive days, so to get at least 2 or 3 full estrous cycles. And we just studied the connection or the link between estrous state and behavior. But we had 3 very important methodological points. The first one is that we studied the same individual females over time, instead of taking different females, different groups of females in different stages of the estrous cycle. And this gives us another source of variation that we can compare to estrus. The second thing is that we decided not to limit ourselves into any specific behavior or predefined phenotype. Instead, we just studied the spontaneous, naturalistic patterns of behavior of mice running around in an open field arena, basically a very large bucket, for 30 minutes a day for 15 consecutive days. The third thing is that in order for us to analyze our data, we used a novel, automatic, machine learning-based algorithm. These are all very fancy words. But what it does is it automatically analyzes behavior, and it gives us a richer, high-dimensional description of behavior that is sensitive enough to capture variability. So the algorithm that we use is called motion sequencing, MoSeq. It was developed in our lab, but has been widely used ever since in multiple neuroscience labs. I just want to briefly explain how it works. So our mice are running around in this open field arena. We record them using the depth camera. So this is 3D cameras. So for each pixel, we have the location and also the height. This is the different colors. We then feed these videos into the algorithm. And what it does is it can automatically take this very long sequence of behavior and segment it into brief, less than a second long behavioral motifs. So that can be anything from turning left to turning right to rearing, freezing, and so on. So now every frame of our movie will going to be labeled with one of these behaviors. So for example, these are a bunch of frames from different mice labeled as a specific syllable. We call this behavioral motif syllables. We think that that's a rear, that's a mouse standing on its hind limbs. And this is how it looks like, boop. And you see that all of the mice are pretty much doing the same thing. And this is a dive. They all sort of like climb down. This is a dart. They all run together. So the algorithm does a very, very good job. And what we end up getting is a data that looks something like this. So we have all of our recording sessions. And for each one, instead of having like 1 or 2 or 3 behaviors, we have dozens of behaviors. In this case, almost 50. And for each behavior, we have this fraction of use or percentage of how much this specific behavior was used in that specific session. And now I'm going to show you the rawest form of the data that I can show you, because I like looking at raw data. And it's basically going to be this table, but I'm going to use colors instead of numbers. So brighter colors are behaviors that are used more, darker colors, behavior that are used less. And I'm going to organize all of our behavioral session, such as all of the sessions that were recorded at the same estrous phase are going to be grouped together. And this is how the data looks like. The resolution is a little bit messed up, but you can probably see it. So to the left, the colors and the letters represent the different estrous phases. And if you squint at this, and you're thinking to yourself, I don't understand what she wants, it all looks the same to me, you'll be right. Because in our data, we really don't find any very clear significant differences in behavior between the estrous phases. And so if we, for example, average all of the behaviors that we see in the same estrous phase to get this mean, behavioral profile for estrous phase. So here, the x-axis is the syllable, so the different behaviors. The y is how much each one of them is used. Different colors, again, the different estrous stages. These lines really overlap. So there's not a lot of difference between the phases. And in order for us to look for structure in our data, we can now represent each session as a dot, just in 2 dimensions. We're going to color it by the estrous phase this session was recorded in. So this is one session. This is all of our session. And you see how the colors intermingle. It means that there's no estrous-dependent structure in our data, or almost no estrous-dependent structure. So if the estrous phase doesn't affect spontaneous behavior, what does? And because this session is called the emergence of individuality, you can guess that what really affects behavior is the individual identity of the female. So I'm going to show you the same exact data, but this time I'm going to reorganize it so all the sessions from the same female are going to be together. And this is how the data looks like. So again, to the side, 1 to 16 is the different females. And you can clearly see two things here. One is that female mice behave very differently from each other. And two, that every single female mouse across session is actually very consistent in its behavior. So it's very stable, it's very similar across the different sessions that I remind you were recorded at different estrous phases. And again, this is sort of like a mean behavior profile for 4 females. You can see how different they are. And this is the same data, but now colored by the individual females. And you can clearly see the structure now. Behavior clusters by individuals. And in order to quantify how much information does behavior have about phase or identity, we can train a classifier that's basically an algorithm. We give it all of our data, and then we give it another new behavioral session, and we ask it to try to guess which estrous phase this session was recorded in and from which female. And if we try to guess the phase, the algorithm does above chance level. So it manages to do it, but only if we average over a few sessions. So we really have to sort of decrease the variability. If we ask it to guess the individual identity, it succeeds in almost 90% of the time on a session-by-session basis. So I'm going to stop, because this is a remarkable level of individualization. Just by looking at the mouse running around in a bucket for 30 minutes, we can tell you which mouse this is. And it's not just 1 or 2 behaviors are different between the different mice. So in this graph, each dot is actually one of the behaviors, and the y-axis is how much information this behavior carries about phase or individual identity. So for phase, we have very few behaviors that are sort of different between the phases, usually running or rearing. But for individual identity, almost all of the behaviors will carry information about which individual this is, which means that we really see this global shift in behavior profile between individuals. And finally, we can directly ask, how much of the variability in our data is actually explained by the estrous phase or by the individual identity? And when we do that, we find that almost all of the variability in the data that we can explain is explained by the individual identity, and almost none is explained by the estrous phase. So I hope that I could convince you so far that individual identity and not estrous phase is what diversify behavior. And now it gets interesting, because individual identity is not a female-specific trait. Males are also individuals, or at least I've been told. Males are, that's fine, we're going to check it. We're not just going to make the assumption. And so if identity drives variability, and men, or males, my apologies, are also individuals, maybe variability in female and male data is pretty comparable. And so we directly tested it by doing the same exact experiment, 16 individual males over 15 consecutive days. And just everybody can come down, males are also individuals. This is, the structure and the male data, and the decoder manages to predict the male identity almost 90% of the time. And now we can look at the variability in the data. And there are a few things that we can measure. For example, we can just look at the entire data set of males and females and ask which one is more variable. The second thing that we can do is to measure the difference between individuals, how different are individuals between males and females. And the third thing is, for every specific individual, how stable I am in my behavior from one day to the next. So this is the within individual variability. I'm going to show you a figure. High levels in this figure is going to be high variability, low levels, low variability. And we were very surprised to find that not only are females not more variable than males, but males were much more variable than females in each one of the measurements that we actually looked at. And males are twice as variable as females in all of these measurements. And so it was very surprising for us, because it sort of debunked this assumption of increased variability in female behavior that really facilitated a lot of this sex bias that we see in research. And we published this paper about a year ago, and we were very surprised to get a lot of responses from other labs saying that they see it in their data as well. And when we looked at the literature, there are evidence that females are not more variable than males, and if anything is true, it's the opposite. So I just want to take a couple more minutes and just sort of summarize that individual identity and not estrous phase, it was diversified female mouse behavior. Females are more stable than males. And so our data really advocates for using both sexes in scientific studies. And actually, it also suggests that if you are looking for a more stable sex to look at, if you're concerned about variability in the data, you should use females and not males. And I keep saying in our data, in our experiment, because variability has many sources that can affect it. We did a very specific experiment. We did it in mice. We did it in a very specific strain of mice under very specific, circumstances. It could be that somebody else will use a different strain or a different age, or even use rats, and we'll get a different result, and that's fine. Because we treat this experiment as a proof of concept for a few things. The first is the danger of misconceptions, and sort of our obligation as scientists to always test our assumptions. And I say we because I was the one who stood in my PI's office and said, we cannot study females. We will not be able to make sense of the data. And he said, really? And I said, well, I don't know. And this is what started this experiment. And for the power of inclusion, because not just from a social perspective, but from a scientific perspective, because exclusion, in this example for females, can not only skew a result, but sometimes hide the results completely, reverse the result. And third, and I think you're going to hear it from the three of us in this session, is about the need to study variability, about the need to unfold the averages, not to decrease variability, but actually to describe it. Treat it as a feature, not as a bug, right? But just because something is variable doesn't mean that it doesn't need to be studied, just the exact opposite. It could be key to understanding, my apologies, what the brain does. And it's true for everything. For example, I used males and females throughout this presentation, and in a minute, Professor Yael is going to stand here and tell you that males and females are also a very variable spectrum. And understanding this variability is going to help us understand how biology works. And with that, I want to thank the people who worked with me on this project. It was really a team effort. And my friend at the Data Lab, and my funding resources. And again, I want to thank you, and thank you, Daniela, for inviting me here. This is so important, and I'm having so much fun. Thank you.